Well, thank you, Purina Institute, that so many great lectures so far, and as always. And I would like to continue the lines of discussions and applications in multi-omics, but we'll shift that focus to cats in chronic kidney disease. I will just give you a very brief background about the kidney and the chronic kidney disease in cats, and then we'll dive into the study and show you the design and some analyses that we performed. If you take a cross-section of the kidney, you'll see two layers. The outer layer is cortex, and the inner layer is medulla. And both layers, they have different anatomy, different physiology, and also perform different functions in the kidney. Each feline kidney contains approximately 200,000 small functional units known as nephron. And each nephron consists of a glomerulus and the long tubules. And most importantly, the kidney is a metabolic organ, which consumes a large amount of energy and oxygen. And as a result, kidney is particularly sensitive and vulnerable for oxidative damage and hypoxia injury. And I will come back to that. So over the many decades, many researchers make many advances toward understanding of this chronic kidney disease in cats. Despite the progress, the molecular mechanism underlying this disease is still incompletely understood. So the goal of this study is try to bring more molecular insight into the mechanism of that common disease in older cats. So Dr. Schneider's talks then show you so many omics technologies, and among all of those, we utilize 3 of them in this study: transcriptomics, proteomics, and metabolomics. And we'll see that in next slides. So this shows you briefly the study design. In the study, we enrolled 2 cohorts of cats. In the cohort 1, there are 35 cats divided into 3 groups: the healthy control and CKD1, 2. We combine the iris stage CKD stage 1 and the stage 2 together because we don't have enough stage 1 cats. And then CKD3, 4 as stage 3 and 4 combined. And the serum sample was taken from, collected from these cats and subject to untargeted metabolomics. And in the second cohort, we have 23 cats, same 3 groups, so it's approximately 8 cats in each group. And kidney samples were collected when cats were euthanized for humane reasons unrelated to this study, and we separate cortex from medulla, and they're both subject to renal tissue transcriptomics and the proteomics. So we did the data analysis integration, and then led to many exciting findings. Due to time constraints, here I will just show you the renal energy metabolisms, and we'll look at the ketone and glutamine metabolisms and hypoxia, and also, if we have time, we can look at the redox homeostasis as well. Let's first look at the ketone. So ketone is also known as beta-hydroxybutyrate. We all know that, in the nephron, we know that different segments of nephron prefer and utilize different energy source. For example, the glomerulus prefer glucose. And the ketone is the preferred energy substrate for the distal tubules. And we know that ketone is produced in the liver and then released to the bloodstream. And ketone then is taken up by the kidney cells by the ketone transporters, here also known as monocarboxylate transporter 1 and then 2, MCT1 and MCT2. Once inside of the mitochondria, the ketones went through a few steps of transformation to become acetyl-CoA, and that can enter TCA cycle to produce energy. So there are a few important enzymes, actually we're going to show 3 here, that perform the ketone oxidizations. OXY1, this is an odd name, 3-oxoacid-CoA transferase. So if you don't remember that, that's okay. So OXY1 is it's a rate-limiting enzyme. And another one, it's ARCADE1, also has an odd name. This is the one enzyme that catalyzes the last step of ketone oxidation to produce ATP. And also there is a ketone dehydroxylase, BDH1. So this enzyme work together to make acetyl-CoA, and then the acetyl-CoA can enter the TCA cycle, produce ATP for nephron. So next slide, we're going to look at the gene and the protein expression change of these key enzymes in ketone metabolisms. So first, let's look at the blood. So we see that we compare the 3 groups to the kidney CKD group with the control, and we found that the ketone level is accumulated in circulation in CKD3-4. And this is the protein expression. The OXY1 is a rate-limiting enzyme. The enzyme protein level is downregulated in both cortex and in the medulla in CKD3-4. Here on the bottom is the gene, renal tissue gene expression. And the bar plot here, the green, gold, and the red bar represent healthy control, CKD1-4 and the CKD3-4 respectively. As you can see that the cortex is in the left and the medulla is to the right. We can see that the gene expression of OXY1 and ARCADE1 are downregulated in both cortex and in the medulla. So our data suggests that there is a ketone uptake and utilization impairment in the CKD, and that this may lead to the ketone accumulation in the blood. So next, we're going to look at the glutamine. So why glutamine? Glutamine is a non-essential amino acid, and that means the cat can make glutamine themselves from the diet. But nevertheless, glutamine is the most abundant amino acid in the blood in cat, and the same is true in humans. And our study found that the glutamine concentration decreased in the CKD group compared to the control. Different than the ketones, glutamine is the preferred energy source for the proximal tubules, as highlighted in the yellow segment there. And most importantly, glutamine play a key role in the regulation of urine pH. So the kidney utilize glutamine to neutralize protons and to maintain the urine pH. So if you don't have enough this glutamine, that you may get an acid-base imbalance. So that's a paper published more than 20 years ago by Jonathan Elliott. His study showed that cats with renal failure has lower urine and the blood pH. So we're going to look at that. So glutamine from in the circulation is taken up by the renal cells by glutamine transporters and transport into the mitochondria. Once inside the mitochondria, glutamine is first converted into glutamate by an enzyme called glutaminase, GLS. And then glutamate is further oxidized to become alpha-ketone glutarate. So alpha-ketone glutarate, Kg, is the precursor of TCA cycle and then can enter the TCA to make ATP. So there are three enzymes that convert glutamate into alpha-Kg. 2 of them happen to be the transaminase. And the one is the GPT2 and the GOT2, and the middle one is the glutamine dehydrogenase. So we noticed that in this chains of reaction, that 2 molecules of ammonia are released when glutamine is converted into alpha Kg. So these ammonia are important buffer for the protons. If you don't have this enough ammonia, so you may have this acidic urine. So we look at the gene and protein expressions here. We already saw in the previous slides that in the serum, the glutamine level is decreased in CKD groups. And we look at the protein expression of these 2 transaminases, GPT2 and GOT2. Both protein expression is downregulated in CKD, in the cortex, and also in the medulla. In the gene expression, there's a downregulation of genes encoding for all these 3 enzymes, glutaminase, GOT2, and the GPT2 as well, in both cortex and in the medulla. So this is an interesting observation. In the medulla, we found that the expression change occurs in both CKD1-2 and the CKD3-4, but in the cortex, that only observed in CKD3-4. So that is a spatial temporal regulation in glutamine metabolism in cat with chronic kidney disease. So our data showed that the glutamine metabolism is impaired in the kidney of CKD, and that may contribute to energy deficiency as well as acid-base imbalance in cat with the disease. So next we're going to talk about hypoxia. This is my favorite topic. So hypoxia actually is is adaptive response to low oxygen levels. So in this schematic of hypoxia signaling pathway, you see in the middle, that's a PhD 1 to 3. So those are not PhD degrees. So they are 3 different forms of the of an enzyme known as prolyl dehydroxylase. So these enzymes are oxygen sensors. So under normal physiological condition, these oxygen sensors, PhD, can sense that oxygen level is optimal, and so they can hydroxylate this protein called HIF-alpha, hydroxia-inducible factor. So once HIF-alpha is hydroxylated, they can be recognized and degraded by proteasome. So in normoxia, there will be no hypoxia signaling is not induced. But when the oxygen level is dropped, possibly due to ischemia, and the oxygen level is low, the PhD can sense that, and then unable to hydroxylate the HIF-alpha. And that stabilizes HIF-alpha, and HIF-alpha then combined to HIF-beta. They are both transcription factors, and they can activate the expression of genes that can induce angiogenesis or production of EPO, trying to bring back up the oxygen levels. So we look at the renal tissue gene expressions. Here in the top is the cortex, bottom is the medulla. And we see that HIF-1-alpha and HIF-3-alpha expression is upregulated in CKD, and PhD2 is downregulated, the oxygen sensor. And the medulla, same thing, HIF-3-alpha upregulated and PhD2 downregulated. So our data suggests that the hypoxia signaling is activated in response to low oxygen in the chronic kidney disease in cat. Okay, so I may just spend a couple minutes to talk about redox. So because kidney consume a large amount of oxygen, so that means that it's very vulnerable to oxidative damage. The oxygen consumption can generate a lot of reactive oxygen species, and the glutathione is very well-known antioxidant. It's synthesized by the enzyme called glutathione synthetase and from 3 amino acid precursors. And then the glutathione cycle between the oxidized and the reduced form to neutralize reactive oxygen species, and that requires 2 enzymes, GSR and GPX. So in that cortex protein expression, we found that these enzymes protein expression are decreased. Glutathione synthetase and 3 different isoforms of glutathione peroxidase are also downregulated. And in gene expression, all the 3 enzymes are also downregulated in the cortex and also in the medulla. And also importantly, in the circulation, the oxidized glutathione accumulate in the CKD group. So this is oxidized glutathione is a biomarker of oxidative stress. So our data suggests that the redox homeostasis is disrupted with increased oxidative stress in CKD. So it's a lot of information to take in. We look at the serum metabolomics and the transcriptomics and proteomics from renal tissues. Our analysis integration that showed that dysregulation in energy metabolism, so we provide evidence to show that the impaired ketone and glutamine metabolism and hypoxia signaling pathway is activated, and also there is aberrant redox homeostasis in cats with chronic kidney disease. So there's a lot other information, more information that in the manuscript was recently submitted. So hopefully that will come out soon. So finally, but more importantly, I want to thank my colleagues across the pond and in the United States, and also my collaborators. Many of them are sitting in this room. I really want to thank you guys to help to make this study possible. So with that, I'm happy to take questions.
Great. Thanks very much, Johnny. Again, we have a couple of questions from a virtual audience, but I encourage you, if you've got any questions, to go back to the microphone. One question is in regard to glutamine metabolism, where the gene expression changed in the medulla in CKD1-2, but in the cortex in 3-4 stages. Have you got a potential explanation for those spatial differences?
Yeah, so the anatomy, renal anatomy, the medulla is inside, and also actually that people measure that oxygen pressure in the medulla and the cortex. The medulla is 2 or 3 times lower than the cortex, so I think that it makes the medulla more sensitive, vulnerable to oxygen level change. So there is also a hypothesis. I think that was a review paper by Sarah Spencer and Jonathan Elliott. They discuss the hypoxia and chronic kidney disease in cat, and they postulate that hypoxia is the cause of CKD, although this is still up for debate, which one is chicken, which one is eggs, but and that will make sense if that is indeed as the cause, because the oxygen level change can actually slight change can impact medulla earlier than cortex. Okay. But that's a hypothesis.
Great. Another question is in regard to the fact that CKD is obviously a disease of aged cats. And so how do you remove aging as a potential factor for explaining the changes that you see?
Say it again?
So CKD is a disease of older cats. Yeah. So how do you remove the potential impact of aging on the results that you have seen?
Yeah, that's a good question. So absolutely, I think that in the 3 groups that their age, we try to balance the age, right? So, but another thing is that in the statistical analysis, we include the age as a covariate to adjust for the age effect. But nevertheless, I think the age, that's always study limitations.
It's hard to find an older cat without some form of...
Because the CKD is always, it's more common in the aged cat.
Okay.
So.
Great. And then there was one other question, which was what are the main similarities and differences between human and feline CKD?
Oh, I think that's another interesting question. So I think they are commons, but also they are very different. The commons in that they both has tubulointerstitial fibrosis, but the cats that happens earlier in cats, and the fibrosis is the hallmark of end-stage CKD in humans. In humans, that a risk factor for CKD is more like diabetes or hypertension, but that doesn't appear to be the case in cats. And in cats, I think some studies are associated with dental disease and vaccination, frequent vaccination. So there's some differences, I think. But
again, thank you very much, Dr. Li.